Hi everyone. So this is I think the second to the last video. I thought I was gonna make only one video na lang pero I noticed na medyo mahaba yung natitira. So I decided to cut it into two. The first video would be from the classifications of proteins based on the function and also the levels of structure, protein structure, and then the last one would be the classifications of proteins based on their shape. And then ang coverage naman no last video would be, bale last na part na siya ng chapter 2. That would be the protein hydrolysis, protein denaturation, and also I added two examples of conjugated proteins. Those are glycoproteins and lipoproteins. So we'll start here with the classifications of proteins based on their function. We all know naman na si proteins, they play a crucial role in almost all biochemical processes. And in terms of their functions, they are very, very diverse. Different kinds of proteins have different functions, which is why there is a separate classification for them. And I believe there are 10 proteins based on their functions. The first three are listed here, the catalytic, defense, and the transport proteins. Makikita nyo naman, from the name itself, well explainable na siya. From the name pa lang siya. So the first one, catalytic proteins, from the name itself, catalytic. So they are proteins that are best known for their role as catalysts. And we all know naman na kapag catalyst, their function is they either speed up or slow down a reaction. So yung mga proteins na to, they have the role of biochemical catalyst. And yung mga biochemical catalyst na yun, they are also known as your enzymes. So yung mga enzymes na to, they participate in certain metabolic reactions that occurs in the cells. They either speed up or slow down this metabolic reaction. So we have a separate chapter for this one, si enzymes, and that would be the next chapter actually, chapter 3. So, ayan na lang. Hindi ko na siya i-expound pa kasi mapapag-aralan naman natin yan on the next chapter. Basta, catalytic proteins, they are also known as your enzymes. And then the next one would be the defense proteins. Well explainable naman, defense. So these proteins are central to the functioning of the body's immune system. And the immune system, it is the system in our body that is responsible in fighting antigens or yung mga foreign molecules or materials na pumapasok sa katawan natin. They are also called as immunoglobulins or your antibodies. I have a separate discussion for this one dun sa last na video, particularly sa glycoproteins, yung mga immunoglobulins and or also known as your antibodies. Basta defense proteins, antibodies sila. And then transport proteins, that's the third classification of proteins. Ito naman, they bind to particular small biomolecules and nagbabind sila or kumukuha sila or they carry small biomolecules and then they transport them from one location to another. Once they reach their destination location, they release that small molecules. Kumbaga, para silang JNT Express sa katawan natin. They transport biomolecules from one location to another. Examples of this one would be si hemoglobin. I'm pretty sure that is already familiar to you. Hemoglobin, it is actually a protein that carries oxygen from the lungs to the other organs and tissues. Ayun. So, yung small biomolecules na dinadala niya is si oxygen. Another example would be si transferrin. This time, this is a protein na it carries iron from the liver papunta sa bone marrow. I'm gonna write it. Hemoglobin. And it carries oxygen, di ba? And then si transferrin, it carries iron from the liver papunta sa bone marrow. So, yun yung transport proteins. They are like the JNT express ng katawan natin. And then, another four types of proteins or classifications of proteins. We have the messenger proteins. Ito naman, they transit signals to coordinate biochemical processes between different cells, tissues, and organs. Minsan, na pag um, parang pareho lang, yun yung common misunderstanding, na parang pareho lang si transport and si messenger proteins. But the difference is, si transport proteins, dinadala niya yung molecules from one location to another. Si messenger proteins kasi is, they only transmit signals para mag-proceed or mag-coordinate yung mga biochemical processes na between the different organs, different cells, different tissues. One example is your insulin and glucagon. So what happens is that once your body um, detects na masyadong mataas na yung sugar level mo, nagre-release si pancreas ng insulin. And then, eto naman si insulin, it would help or transmit signals doon sa mga cells. Tapos, it would help them to absorb glucose. Therefore, it would be able to reduce your blood sugar level. And then, kapag naman si glucagon, once your blood sugar levels are too low, si pancreas magre-release yan ng glucagon. And then, in turn, yung glucagon, it will instruct or it will transmit signal doon sa liver mo na mag-release ng mga stored glucose niya. Diba? Si liver, it stores glucose. So, it releases glucose. Therefore, yung blood sugar mo is tataas or it will rise. So, yun yung difference ni transport and si messenger proteins. Si messenger, it transmit signals. Yan. Insulin and example. Insulin and glucagon. Ayan. And then the fifth one is the contractile proteins. These are proteins that are necessary for all forms of movement. Examples of this one, basta movement siya. Movement nung isang cell or isang body. Ayan. Examples would be muscles. Ayan. Muscles, it is made up of filament-like contractile proteins. And we all know naman muscles, it is needed for movement. And as well as the actin and myosin. And also yung flagella ng sperm cells. Flagella is the parang tail, yung buntot ng sperm cell. And 'di ba, yun yung responsible para makaswim or makagalaw yung sperm cells. Sperm cells. Ayan. Ayan. And then the sixth one, structural proteins. From the name itself, it provides a structure. It would confer stiffness and rigidity to otherwise fluid-like biochemical systems. It gives structure. Examples of this one would be the collagen and the alpha keratin. Ayan. Si collagen kasi, it is a component of a cartilage. And then si alpha keratin naman, it is or it serves as a protective covering sa hair, fingernails, feathers, ayan, mga hooves, and several animal shells. So it gives structure or covering, protective covering. Si collagen and alpha keratin, it will be discussed further mamaya, I think, doon sa may levels of structure and doon sa classification based on the shape. Parang natatandaan ko ginawa kong example si collagen and alpha keratin. And then the seventh one would be the transmembrane proteins. These proteins naman, they help control the movement of small molecules and ions through the cell membrane. So ito ulit, quite similar si contractile and si transmembrane proteins. Both of them would help with the movement. Pero si contractile kasi movement ng buong body or buong cell. Eto si transmembrane kasi is it would help control the movement of the small molecules papasok or palabas doon sa isang cell membrane or cell. Parang nagiging entrance or exit sila. It would help with the entrance and exit of some small molecules and ions. Um, papunta and palabas dun sa mga cell membrane. So yun yung parang pinaka function ng transmembrane. Nasa micro level siya, kumbaga. Nasa cell level siya. Si contractile proteins naman macro siya. Yun yung difference nitong dalawang to. And then the next one, I think that's the last three. Ayan. Si storage, regulatory, and si nutrient proteins. Si storage, it binds and stores small molecules for future use. Ayan. They serve as bodega, kumbaga, storage proteins. Examples would be si ferritin and si myoglobin. Si ferritin, it is an iron storage protein. Dito sa taas na ferritin, iron storage. And ferry, I think that means iron, di ba? The Latin or the Greek word of iron is ferrum. Yan. So iron storage, which saves the iron for the use in the biosynthesis of new hemoglobin. So yun yung parang doon kumukuha ng iron once nagkaroon ng biosynthesis or production of hemoglobin. Yan, si ferritin. Yun yung use niya. Another example is si myoglobin. Ito naman, this is the oxygen storage in muscles. Ayan. And then the second to the last classification would be the regulatory proteins. Ito naman, they act as sites at which messenger molecules can bind and thereby initiate the effect that the messenger carries. Para silang switch, kumbaga. Diba, once it binds with the messenger molecules, and remember messenger molecules, they carry small molecules with them from one location to another. So once they reach the destination, location, yung mga regulatory proteins na to, it will bind with the messenger molecules. Matatransfer yung small molecules na dala-dala ni JNT, ni messenger molecules, and then, ang gagawin ni regulatory proteins, it will initiate or it will activate the effect nung dala-dalang molecule ni messenger uh, proteins. Another one is regulatory proteins when they bind with the catalytic proteins or yung mga enzymes. So once they bind with the enzymes, they would either turn on or turn off or control the enzymatic actions that these enzymes would do. So para silang switch. They either activate or deactivate the enzymes or the small molecules carried by these messenger proteins. Yun yung regulatory proteins. And then the last one would be the nutrient proteins. These are proteins important in the early stages of life, especially from embryo to infant. An example of this one is casein, which is commonly found in milk. And casein is mostly made up of 3/4 of the proteins found in milk. And we all know naman na si casein, it is the nature of this one is to nourish and also provide, ano ba tawag nito? Immunological protection para doon sa mga bata. Mammalian young, either animals, baby animals, or hindi mo sa fetus, babies, ayun, infants, ayun. And then another example is of albumin, which is commonly found in your eggs. Ito yung proteins na galing sa eggs. And yung eggs na to, it is over 50% of albumin. Which is why kapag may mga naririnig kayong nagda-diet and they do not want carbohydrates, they only want proteins, they go with eggs instead of meat. Kasi si eggs, mayroon siyang nutrient proteins, and that's your of albumin. Okay, so for the levels of protein structure, we have four: the primary, secondary, tertiary, and the quaternary structure of protein. So for the primary structure of proteins, this identifies the order in which amino acids are linked together in a protein. So bale, ang sakop ng primary structure is just the order of attachment ng mga amino acids mo to each other through the peptide bonds. So, yung parang arrangement ng amino acids mo. For example, we have this peptide bond of myoglobin, which consists of 153 amino acids. So, for the myoglobin, ito yung N-terminal end mo. It starts with a glycine, and then leucine, serine, and then aspartic acid. And then it ends with, I think that is GLN is glutamine, and then ito phenylalanine, tapos glycine, leucine, and then glutamic acid. Ayan. So, ito yung primary structure. Remember, myoglobin, 153 amino acids. It is the oxygen transport oxygen storage in the muscles. Ayan. And then, ganoon lang, kadali yung sa primary. Basta yung order nung amino acids mo sa isang peptide or protein chain. And then, we proceed now with the secondary structure of proteins. Dito naman, it is about the arrangement in space adopted by the backbone portion of a protein. So, for instance, you have your primary structure na, and then you try to have it a 3D shape. Ayun, or shape in space. So, yun na yung secondary structure. It could be an alpha helix or a beta pleated sheet. Minsan, alpha or beta lang yung tawag niya. Minsan, alpha helix, minsan, beta pleated. So, from the name itself, for alpha helix, the single protein chain adopts a shape that resembles a coiled spring or helix yung shape niya, with the coil configuration maintained by hydrogen bonds. And then for the beta pleated sheet naman, it is a secondary structure in which two fully extended protein chain segments in the same or different molecules are held together by hydrogen bonds. Para siyang pleated sheets siya. You imagine a paper, tapos yung manner of folding mo para makagawa ka ng pamaypay, ayun, may pleats siya. So, yun yung itsura ng beta pleated sheet. So, you'll notice here, both of these are maintained by this so-called hydrogen bond. So, this is the interaction that um, maintains or is responsible for these two types of secondary structure. So, yung hydrogen bonding na to, it is the bond between the oxygen atom doon sa carbonyl group mo and also the hydrogen from your amino group. Diba? Remember your peptide bonds? C double bond O siya, yung peptide bonds mo, di ba? C, double bond O, and then merong N, H. So, yung O mo dito, it will react with the hydrogen of another amino group mo. But this time, yung N mo dito is from the, ayan, parang ganyan. is from the amino group, from the peptide bond of another. Oh, parang ganyan yung tsura niya. Ayan. Ito yung hydrogen bond mo. Ayan. Kung malino ba yan. Basta, I'll show you later. Ayan. Basta both of them are maintained by the hydrogen bonding. Ayan. Both of them. I think I have pictures of them. Okay. Ito. So, this is an example of a alpha helix na structure. So, siya sa alpha helix, ito yung sinasabi ko kanina. You have your C double bond O here and yung NH mo dito. So, bali, ito yung peptide bond mo. Ito. So, yung oxygen mo doon sa carbonyl group mo, it will react to the amino group nung isang peptide bond nung isa pang amino group mo. So, yun yung hydrogen bonding. Ito, this is the um, arrangement of protein backbone na alpha helix without the details. Makikita mo lang na yung shape niya is parang naka-coil siya. It resembles a coil. And then itong second or letter B na drawing, it already shows the hydrogen bonding interactions. So, ito yung C double bond O, ito, tapos meron kang N mo dito, NH, and then you have C, and then R, and then C double bond O, tapos eto ulit, merong N ka dyan, ganun. So, parang ganyan siya. This is already your peptide bond. So, yung C double bond O mo dito, it will react with the hydrogen nung amino group mo, amino acid mo na nasa ibaba. So, yun yung dot 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 dyan. Ayan. So, ayan yung hydrogen bonding. And then, we have another hydrogen bonding here, another one dito. So, parang ganyan yung hydrogen bonding na sinasabi ko. Ito naman, yung letter C dito would show the atomic detail. It would show all of the atoms, like your gray circle dyan would represent the carbon, yung blue, nitrogen, yung mas maliit na blue, hydrogen, and then oxygen, yung red, and then yung green would be your alkyl groups or your side chains. So, for the alpha helix, makita nyo na lahat ng mga side chains nila is directed outwards. Palawas sila. It points away from the long axis of the helix. So, eto, yung letter D dito, this is the top view. So, makikita nyo na lahat ng mga color green dito are your alkyl group. So, palabas silang lahat. And then, yung C double bond O mo and yung NH mo is either pataas or pababa siya. Ayan yung hydrogen bond mo. You have another one there. So, yan yung sa alpha helix, secondary structure. Then we go on with the next one, si beta pleated sheet. Ito yung sinasabi ko na para siyang paper folded in a pleated manner na parang gagawa ka ng pamaypay. Ayan. Meron din siyang hydrogen bonding. We have this one. So, dito naman, yung sa alpha helix kasi, di ba, yung alkyl group mo is palabas. Eto, sa beta pleated naman, either pataas or pababa yung alkyl groups mo. And then, yung C double bond O mo and yung NH mo, yung peptide bond mo, it lies within a single planar or plane. So, for example, eto, makita nyo dito, ayan yung chiral carbon mo and then yung R mo is pababa. So, for example, we have this one. So, yung R mo dito, the same with, yan, kuma. And then, yung R mo dito is the same with that one. So, nasaan yung C double bond O tsaka yung NH mo or yung peptide bond mo? Nandito yung chiral carbon mo, nandito yung chiral carbon mo. So, yung C double bond O tsaka yung NH mo, yung peptide bond mo, it lies in this plane. Ayan. I'm gonna write it. C double bond O, tapos di lang siya kasha, pero basta dyan. Nandito sa plane na to yung peptide bonds mo. So, makikita mo, yung nasa corner o yung parang folds ng sheet na to would be the placements kung nasaan or locations ng mga chiral carbons mo. And then, yung mga nandito sa planes na to would be your peptide bonds. So, yan yung sa um, beta pleated sheet. There are also two kinds of beta pleated sheet, the parallel and the anti-parallel. Si parallel yung pagka-fold niya. Parallel. Beta. So, yung pagka-fold niya is parang ganyan. Diba? You have two sheets. And then, pareho. So, yung arm mo dito, pareho silang pataas. And then, yung arm mo dito ulit, parehong pababa. So, yun yung parallel na beta sheet. And then, we have another one, the anti-parallel. This is the opposite. Wherein, kung yung arm mo kanina is pataas pareho, yung isa pataas, yung isa pababa. So, parang opposite sila. Then, yung isa, ganito siya. So, yun yung, okay. So, parang, ganyan. Yan yung anti-parallel and yung parallel beta sheets. And then, take note na very few proteins lang yung merong entirely na alpha helix or beta pleated sheet yung structure nila. Kasi most of the proteins have only certain portions of their molecules na merong ganitong conformations or structure. So most of the proteins, they have this what we call as the unstructured segments, eto, or yung mga um, protein chains na walang definite shape, unstructured segments. So we have this parang photo or image of a certain protein wherein it has parts of it, meron siyang beta pleated sheet, and then two parts doon sa protein na to has the alpha helix na shape, and then most of it already has an unstructured segment. So, saan mo ba mapapansin kung nasaan yung beta tsaka yung alpha? Ay, beta-alpha and kung sino yung nasaan yung unstructured segment. Kasi yung mga alpha and beta mo na um, structure, yung mga helical or pleated structure mo, they are found in the portions ng mga proteins na yung mga amino acids nila, yung side chains nila, are relatively small. Konti lang yung mga side chains nila. Kaya nagkakaroon sila ng definite shape or structure. So, once malaki yung R groups or yung side chains, nadi-disrupt kasi yung structure na to, which is why nagre-result sila ng walang maayos or magandang shape. Kaya unstructured yung segment nila. So, eto, both of have, these are small R groups. So, kapag unstructured, large na yung side chains nila. So, yan yung difference between the three. And then, next would be the tertiary structure. Ito naman, overall three-dimensional shape of a protein that results from the interactions between amino acid side chains that are widely separated from each other within a peptide chain. An analogy of this one is your telephone cord. Ito. The primary structure for that one is the wire itself. Yung parang kunwari straight na wire muna siya. Yun yung primary structure niya. It contains the insulator and the copper wires ba? Kung merong copper wires doon. So, yun. Yun yung primary structure niya. And then, the secondary structure for that one is an alpha helix kasi it resembles a coil. So, yan yung secondary structure. And then, the tertiary structure for that one is the supercoiling arrangement. Di ba? Once you, parang tumawag ka, di ba, using the telephone. Then, pagbaba mo, coil na siya, and then parang natirintas ulit siya. So, yun yung tertiary structure. Yung may shape na nga siya, tapos nakakreate pa siya ng another shape. So, yun yung tertiary structure. Parang shape within a shape. Yun yung tertiary structure. So there are three interactions that is responsible for the tertiary structure. Before I forget pala, the difference between secondary and tertiary structure is that secondary structure, it only involves the peptide linkages. And then for linkages. And then for tertiary structure kasi, it involves the interaction between the R groups and results from the interactions between the amino acid side chains. Kanina, peptide linkages, yung mga hydrogen bonding mo between the carbonyl and the hydrogen. Amino group mo, di ba? So, sa tertiary, R groups na siya. So, ito yung mga interactions responsible for the tertiary structure. We have four disulfide bonds, electrostatic interactions, or also known as the salt bridges, and then hydrogen bonds, and then hydrophobic interactions. So sa disulfide bonds, I have already discussed that one. It is the bond between two cysteine amino acid residues, just like this one. Diba si cysteine lang yung may kaya na magkaroon ng disulfide bonds. And actually, disulfide bonds, this is the strongest among the four interactions. Ayan. Wait lang. Ayan. Strongest. Ayan. Ganyan lang for disulfide bonds. Dalawang cysteine na amino acids. And then for electrostatic interactions or also called as the salt bridges. Bakit salt bridges? Kasi it involves an acidic and an basic, acidic and a basic side chain. Diba? If you combine an acid and a basic, ang acid and a base, ang mapoproduce mo is a salt. Acid plus basic, mali. Acidic pala. Acidic plus basic R group. Ayan. So, kapag acidic yung side chain mo, automatic yung amino acid mo is a polar acidic. So, kapag basic naman, polar basic. So, the side chains of these two amino acids, they carry charges, di ba? So, tawag nito, for example, this one, si aspartic acid, this is an acid, so it carries a negative charge. So, si acidic, gawin natin dito, acidic, that carries the negative charge kasi from COOH, magiging COO negative na lang siya. And kapag basic naman, it would carry the positive charge. Kasi from NH2, magiging NH3 siya. So, for example, we have this one, aspartic acid, which is a polar acidic amino acid. And then we have lysine, which is a polar basic amino acid. So, merong interaction, electrostatic interaction between your O negative and your NH3 positive. So, yun yung sa electrostatic interactions. And then next would be the hydrogen bonds. This can occur between amino acids with polar R groups. Ayan. So a variety of the polar side chains can be involved, especially those that possess the different functional groups. Ano nga ba yung mga functional groups ng mga polar neutral na amino acids? Those that have the hydroxy and amino group, di ba? So, basta polar neutral, yung mga amino acids mo, they have the tendency to form hydrogen bonds. For example, you have your glutamine, eto. Meron siyang C double bond O, may amino dyan. So, polar neutral yan. And then, eto, si serine, it has an hydroxy group. Excuse me. So, OH. So, there would be a hydrogen bond between your oxygen and the hydrogen galing kay serine. Ganun lang, yung sa hydrogen bonds. And then the last one would be the hydrophobic interactions, or this is the interaction from two non-polar side chains. So, or non-polar na amino acids are groups. Ayan. So, if you have an aqueous solution na mayroong ganitong protein, mapapansin nyo na yung polar R groups would be directed outwards towards the aqueous solvent kasi si aqueous solvent mo is polar in nature. So, ma-attract yung polar, hindi siya ma-attract. Parang mapupunta doon yung polar R groups mo. Yung mga amino acids mo, kung maiintindihan nyo ko. You have your proteins, di ba? And then, lahat ng mga amino acids mo na may polar na R groups or side chains, ang direction nila is outwards, papunta doon sa may aqueous solution mo. And lahat ng mga amino acids mo na may non-polar na side chains, paloob yung direction nila. So, nagiging hydrophobic sila. Ayaw nilang makipag-interact doon sa water molecules doon sa aqueous solution mo, which is why tinawag siyang hydrophobic interactions. So, lahat ng mga non-polar na amino acids, side chains mo, sila-sila na yung mag interact. Kaya, nagkaroon ng interactions. Hydrophobic interactions. Interactions from the non-polar side chains. So examples of this one would be the phenylalanine and psilocin. And both of these are non-polar amino acids. And so magkakaroon ng interaction between your benzene ring dito and also your isopropyl. This is the isopropyl, hindi itong buo. Ito lang yung isopropyl. So, ayan yung hydrophobic interaction. So, the interactions between the hydrophobic interactions is also called as the London dispersion kung na-review nyo yung chemical bonds. And as you all know, London dispersion, they are weaker than the hydrogen bonds. So if you compare electrostatic and hydrogen bonds with the hydrophobic interactions, si hydrophobic interaction yung weakest sa kanila. Kasi si electrostatic and si hydrogen bonds, um, there is an involvement of atoms with high electronegativities, like your oxygen and nitrogen. Compare mo kay hydrophobic na carbon. Carbon-carbon lang naman. Mas mataas yung electronegativities ni oxygen and nitrogen. Kaya, weaker yung bonds ni hydrophobic kesa kay electrostatic and kay hydrogen bonds. And also, if you will notice, lahat ng mga interactions na to, they are quite dependent with the type or the nature of your amino acids. Si disulfide, automatic cysteine na yan. Sa electrostatic interactions, between a polar acidic and polar basic na amino acids. Si hydrogen bonds, polar neutral silang dalawa. And then si hydrophobic, pareho silang non-polar. So, ayan lang. Yun lang yung kailangan nyo tandaan for the tertiary structure. I think I have an example here. Ayan. Identify the type of interaction that occurs between the side chains of the following amino acids and show the interaction using structural representations of the side chains. So first, we have serine and asparagine, and then glutamic acid and lysine. And so for serine and asparagine, we just have to write the structural formula of the side chain. Kasi tertiary structure ito. Ang kailangan mo lang naman is the side chains. So, ayan. This is for serine, di ba? So, ser. And CH2, and then OH. And then for asparagine or ASN, yan yung name niya. CH2, and then, eto. Andito na yung chiral carbon. So, eto, NH2, may amino, and then O negative. Ayan, O. Double bond O pala. Okay. So there would be an interaction between these two. Si oxygen and hydrogen. Paano ko nasabi? Hydrogen bond yan. Hydrogen bonding. Bakit? Kasi si serine, it is already polar, ano ba? Polar neutral siya. And then si asparagine mo is also a polar neutral na amino acid. So automatic kapag polar neutral naman, 'di ba? It is hydrogen bonding. So hydrogen bonding. And then the last one, glutamic acid and lysine. So si glutamic acid, dalawang CH2. You can just shortcut it that way. Then C double bond O negative. And then si lysine, CH2 na apat. And then meron siyang amino group. Ayan. So mapapansin nyo, may negative and positive na siya. Kasi si glutamic acid mo is already a polar acidic amino acid. Polar acidic. And then si lysine mo is polar basic. So the interaction between these two molecules. So this is an electrostatic interaction. So ganyan lang. As easy as that. Diba? Actually, the shortcut for that one is to just identify kung ano yung nature ng mga amino acids na yan, and then malalaman mo na agad kung ano yung tertiary structure niya. So, ayan. And then next would be the quaternary structure of protein. So dito naman, this is the highest level of protein organization. Ito na yung pinakamataas. So organization among the various peptide chains in a multimeric protein. So for example, we have this one. This is an hemoglobin protein. So you have, see hemoglobin, it is a tetramer. It contains two alpha chains and two identical beta-pleated chains or sheets. Ayan. So ito yung beta, beta, ito yung alpha, ito yung alpha. And also it contains a heme group wherein this is the site where the oxygen binds to the protein. And diba remember, hemoglobin is a transport protein. It transport oxygen. So si heme group siya yung nagdadala nung or nagbabind or nag-carry nung oxygen mo. Ayan. So the quaternary structure of proteins. Ito. I think I have this one. Okay, ito. So, ito na yung parang difference between the four levels of structure. So, this is for hemoglobin. So, this is the structure. Ito lang yung parang hindi nilagay lahat-lahat. Kasi mahaba yung sa hemoglobin. I think that's 53 amino acids. So, part of this is a peptide chain taken from the hemoglobin protein. This is the protein structure. Ito yung order of arrangement ng mga amino acids niya. And then for the secondary structure, you shape that. Ang shape niya is a coil. So that is a alpha helix secondary structure. And then yung alpha helix na yan, i-coil mo siya ulit. So magiging polypeptide chain siya. And then yung polypeptide chain mo, you get two alpha helix na polypeptide chain and then dalawang beta na polypeptide chain. You assemble of all of this, yung mga subunits. This is a subunit. So you assemble all of these subunits, you would be able to form your quaternary structure. So yun yung levels of protein structure. So this is now the structure or the core structure of your hemoglobin protein. Okay, then next, Probably the last classification of proteins that is based on their shape. We have two, the fibrous and the globular protein. I think this one, fibrous and globular protein, they are now the shape of their quaternary structure. So, see, fibrous protein, these are proteins whose molecules have an elongated shape with one dimension much longer than the others. And then kay globular naman, these are proteins whose molecules have peptide chains that are folded into spherical or globular shape. So, parang mas magulo yung kay globular shape. It would parang crumpled paper siya hanggang sa naging ball na crumpled paper. So, si fibrous, fibers siya. So, the difference between the two in terms of their properties is that si fibrous proteins, they are water insoluble. Water insoluble. And then si globular protein, kabaliktaran yan, they are water soluble. And then, in terms of the secondary structure, there is a single type of secondary structure. So, kay fibrous, kung alpha helix yan, alpha helix lahat yan. Pero kapag kay globular kasi, there's a combination of pwedeng alpha, pwede rin siyang beta. So, only single secondary structure. And then, kay globular is several types. Dari, structure nila siya kasha. Ayan. And then in terms of their functions naman, yung mga fibrous proteins, they provide support and external protection. Just like your, eto example, si collagen, sina protein, di ba? They are your structural proteins. And then kay globular proteins naman, they are involved in metabolic chemistry, performing functions such as catalysis, transport, and also regulation. Example is si hemoglobin, and hemoglobin is a transport protein. Diba? Lahat ng mga transport proteins mo, enzymes, and also yung mga regulatory proteins mo. They are globular protein in shape. Okay. Ito yung mga examples of fibrous and globular proteins. We have keratins, collagens, elastins, myosins, and fibrin. And these are their corresponding occurrence kung saan sila makikita. And then for globular proteins naman, we have insulin, myoglobin, hemoglobin, transferrin, and immunoglobins. And I have already ang tawag nito, mention them before doon sa based on their function. So makikita yung mga functions nila, they are quite the same. They are involved with enzyme catalysis, transport, regulatory, di ba? Storage, ayan. So yun yung fibrous and globular. Okay. This one is an example of the levels of protein structure for alpha keratin. So, hindi na in-include dito yung primary. So, si alpha keratin, etong letter A is the alpha helix. So, obviously, this is the secondary structure. So, alpha helix siya. And then, dun sa letter B, a pair. You get a pair of these secondary structure na helix. So, a pair of these helices would twine or coil about one another para makaproduce sila ng coiled coil. Diba? You heard that? Coiled coil. So, coil siya, tapos i-coil mo siya ulit. Um, parang natirintas siya, tapos ititirintas ko pa ulit. So, yun yung tertiary structure niya. Then another, probably this is also the tertiary structure. Kasi, di ba, yung coiled coil na to, you get another one of that. and then you coil them again. So, para 3 coil to the third power na yan. So, this is called a protofilament. So, that is a pair of coiled coils na na-coil with one another. And then, you get 4 of these protofilaments, and then you coil all of them. So, makakabuo ka ng microfilament. That is 4 coiled protofilaments. And this is now the quaternary structure for your alpha keratin. And also, this is the core structure unit of, or sorry, core structure of one unit of alpha keratin that is found in our hairs. So, ganyan yung fibrous. Fibrous protein, si alpha keratin na makikita natin sa buhok natin. Ayan. So, this is the secondary, siguro 2 degree. And then, both of these are the tertiary. And then, ito yung quaternary. Quaternary. Ayan. Quaternary structure. Tapos, ito naman sa globular proteins. Ito yung sa hemoglobin. Diba si hemoglobin, it is a tetramer. It contains 4 peptide chains. So, 2 of the peptide chains are alpha helix, and then the other 2 are beta helix. And diba, remember, globular proteins, they contain several types of secondary structures. So, may alpha and meron siyang beta chain. And then, it also consists of a heme group. So, this is the structure of heme group. Since hemoglobin is a transport protein, it carries oxygen. So, sino yung mag-hold ng oxygen during their transport? So, siya si heme group. Yung iron sa gitna ng heme group mo, siya yung nag interact with oxygen or yung O2. Kaya na-carry nila si oxygen from one location to another. So that is the protein structure of your hemoglobin. Okay, so the next, this would be now be covered sa next video, C14 hydrolysis. So that's it for this discussion. I hope you learned something. I hope you took notes of what I have said kasi hindi ko naman isusulat lahat-lahat. And if you feel like wala kang naintindihan, wala kang masyadong maintindihan, you can browse this video again or you can ask for me. Okay? So I hope you learned something and then I'll see you again next time. Bye-bye.